episode 312 of the Stem Cell Podcast, Biomanufacturing in Space, with Dr. Pinar Mesci. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us, leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, joining us, we have Pinar Mesci. She's from Axiom Space, and she's on the show to talk about her work on biomanufacturing in space, not actually her in space, but how we can get to that point where we get efficient biomanufacturing in space. It's going to be a great chat. We've also got our usual roundup of recent highlights and stem cell news that's coming right up in a few minutes.
But first, registration and abstracts are open for the Global Stem Cell Event, the ISSCR 2026 annual meeting, July 8th to 11th in Montreal, Canada. Join nearly 3,500 scientists, clinicians, and industry leaders for 4 days at the frontiers of stem cell science, regenerative medicine, and engineering biology. From fundamental discovery to clinical breakthroughs, this is where collaborations start and new ideas take shape. Submit your abstract by February 25th for a chance to present your work and be considered for travel and merit awards, and design your dream session and submit a science spotlight proposal by February 25th as well. So be part of the global conversation shaping the future of stem cell research. Visit isscr2026.org/scientific-topics, where you can sort the agenda by theme, and topics include the stuff that we're going to be talking about, of course, bioengineering, organoids, neural stem cells, aging, and more. We got a lot of cell stem cell papers today. Actually, nothing but cell stem cell papers, 4 for 4. And we're going to start things off with a familiar topic, and a topic that's very familiar to Daylon, obviously. This is a stealth stem cell paper titled Development of Human iPSC-Derived Ovarian Support Cells as a Clinical Grade Product for In Vitro Fertilization. And full disclosure, this is work that we've already kind of talked about with the folks at Gamito, this startup company led by Dina Radenkovic, as well as the scientific leads, Bruno Paulsen. We actually had an episode chatting with them about this product for Tilo a few months ago, a year ago, more than a year ago. But regardless, check that out for the appendix to this particular roundup paper that I'm going to be covering. And you know, it's IPS applications for reproductive biology and fertility, and certainly we've talked about iPSCs all the time here on the show, and they show promise in the development of different strategies to alleviate reproductive diseases. That's really the focus of what Gamito, this company, is all about, and they're hoping to improve reproductive outcomes as well. So here they're showing some data from the clinical development and application of their ovarian support cell product, or OSC product, that we talked about on that interview for Tilo, to improve the ex vivo in vitro maturation of human oocytes, right? And first they showed that the science behind actually making these OSCs, ovarian support cells, from the iPSCs was pretty robust. It's a transcription factor mediated differentiation process that can ultimately, in a co-culture system, improve the oocyte maturation rate. So, but their bigger vision is towards broader clinical applications, as we'll talk about in a little bit, where they talked about different materials that they use in the process and the generation of clinically suitable iPSC master banks and cell banks. They did a lot of deeper transcriptomic analysis of the resultant ovarian support cells created from these iPSCs, showing the things you want to show: reproducibility, consistency across different batches. And then they did a analytical release testing and development of a mouse oocyte maturation assay. I think that's something they developed in-house to actually to assess the potency of these ovarian support cells as a product, this Fertilo system as a product, right? And then they did the in vivo, not in vivo, but the human applications here, which is the application of Fertilo in a longitudinal cohort analysis, showing improvement in different outcomes when it comes to the maturation of the oocytes compared with traditional in vitro maturation principles here. And they're showing the clinical development and application of this iPSC-derived product to hopefully improve successful reproductive outcomes. That's the ultimate goal here, and as we know that this is a really big market that they're hoping to tackle. I know you have obviously a strong background in this field, Dalen, so I'm sure you got a lot to say here.
Yes, and I mean, I wanna, start by saying I really admire the ambition of the scientists and, leadership, behind this study, and I think it's a really great example of how quickly and efficiently, I should say, not necessarily quickly, although at pretty rapid pace, but, efficiently you can bring this kinda proof of concept in a scientific lab to a you know, clinical trial and potentially commercial marketplace. Um, so yeah, this is a great example of this systematic approach and all the elements, kind of almost like a handbook for anybody who once has a similar idea. And I'm sure the pace was facilitated by the major investment that they were able to recruit in support of this technology. And I think it does a good job of addressing a clinical need out there, which is explicitly stated in the paper as, patients that are at risk of ovarian hyperstimulation syndrome, or maybe PCOS patients, or patients undergoing fertility preservation, all of which may have to undergo this abbreviated stimulation, ovarian stimulation, instead of, a week to 10 days, with the latter half being that the most uncomfortable phase there. You could do this like mini stim, abbreviated stimulation, and minimize the risk for those patients, or get them into therapy, chemo or radiotherapy faster, of course, to address, cancer or whatever it is. So I think if that were the whole story, I would have zero qualms. I don't want to be critical, and this isn't necessarily a critique. I think it's more opportunity for like us all to reflect on how we want to reach the end game here, and really being very specific. I would suggest, and I'm sure maybe the terminology is a little bit pejorative, but I think conceptually, even the Gamito people agree this is kind of a Trojan horse, in that the unmet need here that's addressed is part of the story, but that vast market of consumers, women undergoing fertility preservation for so-called social reasons, that's what we're really talking about here in terms of who's going to adopt or choose this therapy. And I think with that in mind, I have a little bit of anxiety, I don't know, cynicism about the confluence of like business interests and clinical interests. I think that we all need to think about that as we ponder bringing our technology and our innovation to market. I think in these early days, the optics are really important. And, there's a lot of opportunity for self-inflicted wounds here in terms of setting us back, either in terms of perception or the regulatory apparatus surrounding translation of cell-based therapies. And what I mean specifically again there is this idea that while it was very efficient in how this kind of the foundational work for phase 3, which is where they're at now, was implemented, but it was quick, it was careful, and systematic, but the pace, it gets me feeling almost like this is less of a clinical endeavor than it's like a VC-backed startup endeavor. And I get concerned about the priorities here, because if the priority is an exit, which is like the standard for whatever IPO unicorn from an investment standpoint, if the the goal is an exit, I wonder where does that leave a bunch of patients who are choosing this therapy for social egg freezing, the majority of whom won't come back, perhaps, and those who do will come back in over a decade or more, decades in some cases, to use their frozen eggs. So yes, not to mention, and I have to say it, that the comp here of IVM, in vitro maturation, to in vitro maturation with ovarian support cells, I think we need to do another comp, which is in vitro maturation with ovarian support cells to like a normal stimulation cycle, because I think that's important. That a woman should know, or a patient should know, that they may be getting less eggs or less quality eggs than they could get by undergoing a full cycle. And of course, that's all going to be in the, they're going to be informed of all these things. But I think as we're laying the groundwork here, I think need to be very transparent about what we're offering to the public here, especially when I really do believe, and I think Dina would say it, they're going after this big consumer market of young women who are undergoing fertility preservation for non-medical reasons. So that's my spiel. But again, I want to finish by saying this is a really impressive demonstration of how you can get these proof of concept studies into people and into phase 3 very, very efficiently and effectively, systematically, carefully, and successfully.
Yeah, I think a lot to unpack there, but like I guess the emphasis for this particular show is always, of course, about the science, and the science and that is represented in this paper looks really sound to me, and it's definitely a landmark for the IPS slash reproductive medicine field. And, I think your discussion is valid, very valid. And it's something that's not just exclusive to what Gamito is doing. Every single startup has to think about these things, right? Startups, of course, have to balance that societal impact of whatever therapy they're developing, the importance of having very sound science as foundational for the therapy. And of course, because by definition they're a company and by definition companies need to make money, they have to think about a return on investment, right? And so, of course, Dina and the leadership wants to have a large market, and they're gonna aim big and target that very large market because their funders, their VCs are, of course, enamored by that and see that as a huge potential return on their investment. So, I think it's a broader discussion that we could have. And as academic scientists, we're not completely devoid or removed from some of these financial discussions as well, right? There's always money in science. That's just the nature of modern science. There's whether it's grant funding, whether it's VC funding, these are all the realities of doing modern science. So I think we would love to have the folks at Gamito back on the show perhaps to talk a little bit more about this. But regardless, I think from a scientific perspective, this is a really great study to see.
Yes, agree with you 100%. I mean, we're all kind of in that position, right? Trying to get our ideas into the marketplace. I mean, we talk about getting our ideas into people, our therapies for disease, but like really, we just want something we do to be of value. And in this case, it's what the market will bear. And again, yeah, the science is solid. Moving on to a couple of stories I'm going to do back to back here because they have this both nano engineering theme. And yes, we're all cell stem cell. I guess over the break, we had some catching up to do. But all these stories, I think, are complementary in that they showcase cells and therapies coming to the market, and overcoming some of the challenges that stand in the face of that. In this case, we're talking about hematopoietic stem cell therapies, my favorite. And this is really complementary and I think kind of a companion to the other big development, arguably bigger development of gene therapies, which are, in the same vein as cell-based therapies that we talk about, curative, one and done, right? A paradigm shift. And in the case of gene therapy, and particularly with hematologic disease and conditions, it's proven, right? We have a bunch of diseases that have been cured, sickle cell perhaps being the most prominent and established application of like CRISPR based technologies, but many other therapies before that, lentiviral, all kinds of gene delivery mechanisms in blood cells. Uh, but there's a lot of challenges, and we just talked about this actually with Deepak, which was such a revelation to me. You should check out that interview because it was deep and broad. And the key takeaway with respect to this story was that like these therapies are out there, they're effective, they're curative. The unmet need is huge, but like they're not being widely adopted because insurance, right? They're expensive. Who's going to sponsor this? So one of the key goals, obviously, is to lower the costs, right? And to make the treatments more effective or efficient. But the problem with the hematopoietic cell therapies is that this ex vivo culture period that you need to target the cells, not just with like the engineering itself, you're doing the gene targeting, but also the cell culture introduces DNA damage, oxidative stress, and that can undermine the self-renewal potential, can cause differentiation of the cells, can lead to loss of that, precious primitive hematopoietic stem cell population that's going to end up carrying on the production of blood in that patient for the rest of their life, right? So the ex vivo culture can diminish the quality and the yield of those cells. And that can ultimately lead to acute effects like the delayed hematopoietic reconstitution, and also these kind of latent or occult effects like shrinking the clonal repertoire that may lead to reduced efficacy, but also like this long-term risk of CHIP, clonal hematopoiesis of indeterminate, potentially even leading to malignancy. So there's a lot at stake, right? And it's pretty well established now that you need to have multiple inputs, right? There's a whole suite of chemical cocktails and additives and cytokines that have been applied to optimize the expansion and retention of hematopoietic stem progenitor cells. But a lot of that, most of that's in 2D, right? And 2D cultures not at all like the physiological niche, right? So this is a story from the group of Raffaele D'Amico, who's at the San Raffaele Scientific Institute in Milan, Italy. Must be tough. You in California, Raffaele in Milan, here I am, freezing my ass off in New York. And I'm in my office, Arun. It ain't right and it ain't fair. Anyway, moving back to the story. This is like an ex vivo, I mean, what a beautiful story. I'm at a loss for words because I love hematopoiesis, but also this is seemingly like a simple idea, but applied beautifully here. They use these things they call nycoids. I don't know what the etymology of the nycoid is, but it's this 3D culture substrate that has like a miniaturized architecture of 10 to 30 by 15 micron units that they make by 2-photon polymerization. So very precise, reproducible, and they culture hematopoietic stems and progenitor cells in this 3D niche, and show that it improves the multilineage differentiation and engraftment, and these are cord blood stem cells. But then they show like mechanistically that it really is about the mechanobiology by multiple measures. And then they incorporate the genetic engineering using all different types of targeting approaches, classic long-range gene editing, also base and prime editing. They even go old school with lenti, um, and show that they can get an increased clonal output and persistence of these targeted cells in xenotransplants. So, like, yeah, I mean, this isn't like a conceptual, blow your mind type story, but I am focusing on these engineering approaches because I really feel like there's a lot left to do on that front in order to translate the therapies that we're all hoping for, right? The biology, we've come so far in the biology. I think now it's time to really make strides on the engineering, and on this case at the nanoscale. They're doing that. Rafaela D'Amico's group, and the next story I have as well is applying these nano engineering approaches to more closely approximate the niche. And I think that's while it is more prominent and highlighted nowadays, I think still understudied and underappreciated.
Nycoids, huh?
Yeah.
Nycoids? You didn't get the connection?
I don't get it.
Nicheoids?
Oh, boy, now I feel like an idiot.
It's okay. Maybe they should have put the E in there. So yeah, it took me a second to figure that out too. But yeah, it's actually nicheoids. I believe that
Oh
that's
my
what they're going
goodness.
for.
I was like,
Yeah.
"Oh, nicotinic? Is there something about nicotine?" Really getting old.
Hey man, it's okay. It's not your fault. Yeah, they missed the E there. Regardless, yeah, I think a really nice application of engineering biology here to take, some of these HSP studies to the next level. I mean, they mention it. There's whenever you do these, transplantation studies, there's a lot of variables that you introduce there. They got to do like the injection of matching number of HSPCs, different settings, and there's just a lot to consider there when it comes to xeno studies. I think to really look at the scalability as well, and also if you're talking more about these nicheoids, they need more refinement in themselves, further optimization to support their scale up, the incorporation into like GMP compliant systems, because ultimately that's what you want to get for clinical applications, right? But hey, still another cool oid to think about. I've never considered a nicheoid or a nicoid. Maybe they are going for nicoid. I don't know. Maybe they pronounce it a little different over there in Milan. But yeah, I think a cool bioengineered study, and I know you're going to be talking about another one pretty soon as well.
Yes, I should have caught that, but I don't know how they pronounce that over there in Milan. Something like niche-oid. But regardless, I think this story and the one I'm about to talk about are emblematic of how we can approximate the niche to great effect. And sometimes that's the barrier to success, in fact. And more big picture, I don't want to go too deep, but this idea of the occult nature of the deficiency in these cells, that makes me nervous, not just in hematopoietic stem cells, because you put them in the body and then you wait and see, right? And you get the immediate measures and the patient's cured, but like, what do you know about what's happening 20 years down the line? And I wonder about that for just all cell therapies. I've kind of hinted at this before, but we got to do better in supporting the cells in an ex vivo environment that is more like the niche, in order to preserve that kind of native and physiological healthy DNA, whatever epigenome. We got to keep the cells like they were in the niche. Going on to the next nano story. This is a great one because it's kind of like opposite. It's like kind of super physiological and from a repair standpoint. And it's also kind of a callback to that first target of, spinal cord injury. It was one of the most vivid examples and mobilizers of fundraising and attention towards stem cell therapies, right? Because it's uh, very visible. You get this irreversible motor impairment, which arises from the death of neuronal and glial cells, but then also the scar, right? The scar blocks axonal regeneration. And even though there's like limited intrinsic regenerative capacity, there's something there. but that scar makes it impossible to bridge the gap. And there's been a lot of thinking along the lines of biomaterials and biomaterial based approaches for bridging the gaps, those lesions, in order to restore the connectivity. Like, just as an idea, conceptually, it's very easy to envision. You just need to like hook the wires back up, right? Um, but the problem is that the substrate that you use there, whatever kind of bridge you're building, has to emulate that soft, hydrated characteristics of the native spinal cord tissue, right? And it also has to deliver the cues, both the cytokine cues, but also the topological cues and the structure that directs neurogenesis, cell migration, synapse formation. Uh, there have been a lot of work using uh, collagen-based scaffolds because they're biocompatible, and you can get these aligned fibers in the architecture of the collagen scaffold, which is good for aligned neurite growth, right? And you can get enhanced neurite outgrowth. But the key limitation with collagen is that mechanically speaking, there's a mismatch. The elastic modulus of collagen is 10 kilopascals around, whereas the spinal cord tissue natively has tenfold less, about 1 kilopascal. And that leads to this post-transplantation structural instability, disruption of the alignment, and inefficient infiltration of those transplanted cells within the graft. Now, there's hydrogels, right? They offer advantages because the elasticity, viscosity is tunable. You can get kind of just short of 1 kilopascal all the way up to 5 kilopascals, and you can inject these things. But conventional hydrogels, they lack the 3D architecture that's essential for directing axon regeneration. And then there's a host. I mean, the intro on this paper was deep, and I learned a lot. Biomaterials, we got to think about this because the biology is only half the battle, as I've been alluding to. Um, but there have been a lot of innovations using microfibers, this extrusion-based bioprinting, a lot. But all of it has limitations of one sort or another, and certainly not at a level where you lead to structured repair of the spinal cord. So that leads to this group here from the Chinese Academy of Sciences. It's led by corresponding author Qi Gu. Um, and what they developed here is the acronym NEAT, which stands for Nano Engineered Extrusion Aligned Tract. You see there's a acronym that I can easily understand and pronounce. NICOID. This NEAT, it's a biofabrication strategy that integrates this shear induced collagen alignment, like it's kind of like an extrusion process that's also mixed with 3D bioprinting, kind of beyond my understanding, to be honest, all the mechanics of it. But I understand the effect. They also use this norbornene functionalized collagen that it replicates the multi-scale fiber organization of the spinal white matter. And they apply this in a rat model with complete spinal cord transection and show that it works. You get robust axonal reconnection, synapse formation, and locomotor recovery, significant recovery of function in these mice, which is always the most vivid illustration of success in these models. I would invite all of you to have a look. It's inspiring because you can actually see cell therapies leading to a clear and recognizable benefit for, in this case, a rat. But I think that, scale notwithstanding, the approach here is complex, nuanced, a lot of details that were beyond me. But as I said, the proof of functional improvement is really an inspiration, and I think I wouldn't be surprised. I know it's been who knows what 25 plus years, but I feel like it's near time that we see these approaches in large animal primate models and start to see some improvement of function. So I'm excited to see that coming down the pike, Arun.
Yeah, that's I think ultimately where these things are going. This is a neat study. See what I did there? It is yeah, I think just ha ha ha. It's a nice application of bioengineering approaches, and I think all of the studies we're talking about today have some element of that. It's not just the pure biology, it's using engineering to take that biology to the next level. I mean, here, this, like you you're alluding to, has been an ultimate dream in the stem cell field for more than 2 decades now. And certainly there are things that they gotta still work on here, right? Like looking at the full circuit of functional recovery after spinal cord injury. It's a systemic thing that they got to evaluate here. And even though they demonstrated the better alignment of the axons in this scaffold, they and like other stuff too, like the synaptic marker enrichment, the myelination, behavioral improvement, which, like you said, was very striking. I mean, they didn't perform mapping of, like I was talking about, those long range relay circuits, like the whole bodily level circuit, including the identification of different spinal neuron subtypes, their like long duration, long extension projection targets, or I think in a nutshell, they got to do a lot more, like a lot deeper connectivity analysis, which I'm sure they're going to look at. So I think more to come here. They got to do a lot more transcriptomic profiling as well, to really get a deeper dive into how this functional improvement is actually coming about. But like what you're saying, and like what I'm going to talk about in my subsequent study, it's the visual, right? It's the visual of the functional improvement in these animals, and in my next study, it's the ability of retinal organoids to actually sense light and respond appropriately, just like on the cardiac side of things, right? It's the functionality of the cardiac tissue, which is improved before and after whatever stimulation. So, sometimes videos speak a million words.
Yeah, for sure. And you laid it out. There's a lot needs to be done for sure. I get excited. I guess I'll have to wait to see, someone getting up out of the chair. But like most things we talk about on this story, these incremental advances are actually like pretty great strides. And as we're seeing more and more, they're building toward this crescendo of clinical realization. And the story you're about to talk to is, again, a vivid example of that. So, by all means, take it away.
Yeah, and actually, just one last thing on the spinal cord stuff. I don't know what you think about this, but I just feel like as of the last couple of years, there have been so many innovations in this area, and I'm not even talking about like the bio side of it, just the pure engineering approaches to alleviate spinal cord injury and the downstream of that. It just seems like every other week there's some high profile paper coming out on this topic. So like we've talked about a lot here on the show, sometimes it just takes multiple shots at the goal to ultimately score, right? And if we score in this area, it's going to be a big win for a lot of people. So last paper, and then once again, 4 out of 4 on cell stem cell here today. Shout out to you, Sheila Chari, if you're listening in. You're welcome. So this is a retinal ganglion cell survival and functional maturation in transiently vascularized human retinal organoids. So I alluded to this. We're taking, retinal organoids, which have some ability to sense light, which is super cool, and then improving them using vascularization. So that's in a nutshell what they did here. Retinal organoids are emerging rapidly in human biology and stem cell biology to model retinal development and disease. And there's a lot of retinal degeneration diseases that can be studied using these model systems. But problem with these organoids, as the problem with a lot of organoids, is the absence of different vascular networks to make them more complex and also stable axonal projections, like what we were just talking about. The stable axonal projections would contribute to long-term survival of those retinal ganglion cells, and improve in their functionality. So they wanted to address some of these issues to improve the functionality, the viabilities of retinal organoids, and to do that, they incorporated stem cell-derived endothelial cells, another one of your favorite topics, to induce a transient vascular-like network in these organoids, and also coming back to the engineering, they use microfluidic devices to actually stabilize the growth of the axons in these organoids. So after they did all that, the engineering approaches and the vascularization approaches, the resulting organoids were better. They showed reduced hypoxia, which is a sign that they're perhaps healthier. They had a bigger size, also a sign perhaps that they're healthier, decreased apoptosis. And uh, all these things indicate long term survival and maturation of the retinal ganglion cells. And they integrated this. I thought this part was really cool. They integrated the organoids with a microfluidic device that actually had microelectrode arrays built into it, so you could actually do electrophysiological analysis simultaneously with the growth of these organoids. In fact, it's something that we're trying to do with some of our organ chip systems here at Cedars. I mean, it's much easier said than done. Let me say that, but they're able to do it, and they're able to enable stable recordings of spontaneous and also optogenetically evoked activity in these retinal organoids, which is was persistent even beyond the time when those control organoids lost their function. Um, and then at later stages, when these things got really mature, the vascularized organoids actually were able to be responsive even more to light, right? So they displayed a photoreceptor driven on off, kind of on off light switch. So really cool in just indicating the highest level of maturation in these things, and also indicating circuit level retinal activity, right? That's what it's all about, to drive the functionality. So we're talking about bioengineering on this series of roundup papers, and this is just another amazing example of how bioengineer approaches can take your organoids in this example to the next level. This is a really useful, I think, long-term functional model of the human retina, and human specifically, because these are pluripotent stem cell derived cells, right? And it could be a really useful tool for, studying diseases of retinal degeneration, developing new drugs for treating eye diseases, you name it. I think there's a lot of potential here.
Yeah, it's so cool the firing to test the function. That was new to me, and I love it. I love the idea. You make something it can see. I mean, I don't want to take the headline away because that's a little bit sensational, but such a cool story. I mean, the one thing that I love the EC thing. So the first thing I went in on is I wanted to see the ECs, and this was as an aside. This was so cool about everything nowadays in science with single cell is every paper I read, I'm like, okay, this is they got some ECs that they made and then they put them in a retinal organoid, and like that question of chicken or egg, are the ECs going to be influenced by the retinoid or vice versa, or how? I wonder about that. And you say, okay, let me just go into the seq data set and pull out those ECs and have a look at them. That was my thought process. So first thing I went to was to look in their plots of the single cell to find the ECs, and they weren't there. And so I assumed that they had just trimmed them out because it wasn't the focus of their analysis. But then in their discussion, actually, they acknowledged that they didn't find the ECs, and this was at 18 weeks. So I mean, understandable. It's a distal time point. Um, but two things about that. One, you got to wonder, like these ECs in vivo, they would have stuck around and formed vessels, right? In addition to their angiocrine influence. Um, but the other thing I wonder is, like, we talk about the complementarity of the biology and the engineering. And sometimes, like, you can have an engineering input that isn't even really necessarily the physiological state. And hear me out here, like I'm sure, and the deals play a role here, but in vivo. But in this study, they add the ECs with that rationale, and they do something. Clearly, they make them better, as you said. But like them sticking around like they do in vivo isn't necessarily a prerequisite to that benefit. So sometimes you can have an engineering approach that's like a hack. And as long as it does the job, it doesn't really matter if it's physiological or not. So I like that as an illustration of the kind of the old box quote, all models are wrong, it depends on if they're useful or not. And in this case, whether the endpoint is modeling the native eye and all the processes are just some, or generating cells that can be used therapeutically, you don't necessarily have to have the exact mirror of the physiological system. You just got to try and incorporate the elements that are essential to get past these kind of go no go points in development. And here, I think Shaheen would be very pleased to see clearly that there's some kind of paracrine influence from these ECs that's facilitating the advance of these organoids in the absence of any kind of like vascular perfusion or blood flow. So, interesting observations on all sides, and a great close, I think, to illustrate the complementarity of the biology and the engineering, and how we're really moving forward on the latter half of that in the current state of the art.
Yeah, it's a good point. I mean, they didn't stick around, these ECs, so they got to be doing something early, angiocrine, paracrine signaling, to really make things happen. I mean, the other possibilities, the ECs just kind of transdifferentiated into something else, like which ECs are known to do, and that's maybe why you're not able to detect them long term. But yeah, I think the experiment that you're proposing sort of it seems like an obvious next step. You take these things and then you put them in like a mouse model, right? To see how the innate in vivo vascularization can actually perhaps integrate with that theoretical vasculature of the retinal organoid here and see if this it can take the maturation here to an even higher level. Maybe it's something that they're thinking about. So I think a lot to do still here and deeper analyses of kind of what these ECs are really doing. But I mean, if you're looking at the functional outcomes, it's hard to argue with that.
For sure. And as you say that, the pace we're moving with these engineering stories, it may happen. This holy grail that we just recapitulate vascular perfusion in vitro one day, or I don't know. Sounds crazy to me, but the way we're going, I shouldn't be surprised by anything anymore. Like a lab in space, for example, which we're going to talk about in just a minute with Pinar Mesci. Before we get there, quick message from STEMCELL Technologies. Take your human pluripotent stem cell cultures further with M-Teaser Plus from STEMCELL Technologies, the most widely published medium for feeder-free human ES and iPS cell maintenance, is now formulated for enhanced performance and versatility. M-Teaser Plus reduces medium acidosis for more stable cultures all weekend long. To learn more, visit www.stemcell.com/mteaserplus. All right, you guys, joining us on today's episode, we have a special guest in, I wouldn't call it a niche industry because it's certainly expanding. Arun could tell you about that. But we have a guest whose expertise is out of this world. Joining us, the global head of biomanufacturing and biotechnology at Axiom Space, we have Pinar Mesci, who is currently, as I said, global head of biomanufacturing and biotechnology, where she's in charge of a portfolio focused on biomedical research programs, including stem cells, tissue engineering, disease modeling, cancer research, and DNA-inspired nanomaterials, part of a broad suite of initiatives at Axiom Space. Pinar, thank you so much for joining us on the show today.
Thank you so much, Dylan. Thank you so much, Arun. I'm very excited to be here.
Yeah, this is a long time coming, but thanks for being here, Pinar. Obviously, we go way back, and we actually have some projects, as folks may or may not know, that we're doing right now. I mean, it's been great to get to know you over the years and work together, of course, in the space biosciences field over the years. I mean, you have such a unique background, and we wanted to highlight that a little bit. That spans, brain organoid research, nerve developmental disease modeling, and of course now space biology at Axiom. I know your story, and I've been so glad to get to know you over the years. But could you tell our listeners how you got here? Like, how does a brain organoid biologist get to working for a space company?
Yeah, thank you so much, Arun. Yeah, it's been a delight working with you guys and in this field that I never thought in a million years that I would end up in. And I have a pretty wild imagination, but so yeah, like you said, I did my PhD in France in neuroscience. I was mainly focusing on ALS mouse models and trying to understand more how we can regulate neuroinflammation and find new therapeutics. And then when I finished my PhD, well, stem cell field was already there. The iPSC technology was up and coming. It wasn't as broadly used as it is today, of course. So this is back in 2013. But then quickly I realized I needed to expand my skill set a little bit from mouse models to iPSCs. So this is where I actually joined Dr. Alison Motri's lab at UC San Diego that I know that you guys did an episode with as well. So I started working in his lab in 2014 using iPSC models. We were trying to model different types of neurodevelopmental diseases, so like autism, different forms of autism. I was mainly interested in immune cells of the brain, the microglial cells. We have also worked on Zika virus, coronavirus. And then one of the projects that kind of started in the lab around 2017, 18 was this very, very side project back then that no one really was working on was about understanding how microgravity can impact neurodevelopment. So Alison had started that project, but yeah, as I said, like no one was really actively working on it. And I was like, Alison, can I please start working on it too? This sounds really fascinating. And so that's kind of like how I got introduced to the microgravity field and the space biology part. So we've done a number of missions to the International Space Station, mainly using automated systems where we grew these brain organoids and then we received them back when they got back to Earth and then tried to understand how microgravity was impacting the gene expression and so on. And then, so this was, as I said, initially a side project. I still had other projects. And then I was continuing academia. Academia has been basically the only thing that I've ever known in my life. My family, there's a lot of academicians, and then everyone has been at the university. So I was never really thinking of going into industry. But after COVID, I don't know. I think I was starting to feel a little bit stuck in academia, unfortunately, even though that's all I ever wanted. And then this is where I think it's really important to have network with different people and continue doing networking. Because one of the folks that we were working with on the space side, so at that time we were working with Space Tango, Jana Studemeyer and we were still keeping in touch. She eventually moved to Axiom Space. And then we were just having these discussions very like over coffee over a couple of months. And then she was like, well, maybe, you can switch to the space field, because in the space field, traditionally, as you know, Arun as well, very well, traditionally, people like you and I, they don't really work in the space industry. It's mainly engineering background. But she was like, okay, I mean, you can know all those parts, but I think it's really important to have someone in the space field that knows really the biology and the stem cell biology, especially. And I was actually very intrigued because again, like industry was never something that I thought that I would be interested in. But this was certainly something that was out of this world, no pun intended. Yeah, basically, eventually I did a couple of interviews and then switched to Axiom. I got to say that initially it was, I think, even for the field, a brand new idea of having a stem cell biologist in this field. So they were not even sure initially that if this could be a good fit. But I think this is where as biologists, we should really leverage what we already know how to, because we're doing all types of problem solving, multiple projects at a time, and all these skills are actually transferable to, of course, to the space field. So this is like a quick piece of advice to everyone because all the skills that we learn here are transferable to the space field as well. So that's kind of how I ended up at Axiom Space. And now it's been 3 years and 8 months that I've been here. 3.5 years, maybe not quite 8 months, but yeah, and then we're still going strong.
Yeah, I mean, a lot has happened in those 3 years, and in that synthesis, there it occurs to me that that the pace of stem cell research and biology and even translation over the last decade has this agreeable and synergistic symmetry with the development and sophistication and capabilities of our spacefaring tech. So it kind of makes sense there that they would operate hand in glove and kind of co-develop. And yeah, I mean, Arun, of course, he's deep in it, but for me, relatively new to the field, have a superficial understanding. But I went deep into a rabbit hole on the Axiom site, reviewing the four missions that have been completed to date. And the first takeaway I had was the volume and diversity of biological experiments that have been or are being conducted in low Earth orbit. The other thing I came away with was this emphasis on establishing this commercial, private commercial foothold in space or in low Earth orbit, at least. Do you think we're still, I mean, you just talked about how it is relatively new. And of course, yeah, you've been there 3.5 years, but that's still kind of the infancy of where you hope to go. And I'm sure a lot of the science is still in early days, right? But it felt like, looking at the website, that we're kind of pushing past the discovery phase into, commercialization being the near-term priority of some of this spaceborne tech. Is this coming sooner than I think, or do you think we're still really firmly rooted in the discovery phase?
Yeah, great question, Dylan. I think, really having commercial space companies in this field now, it's really gonna propel how we do things in a much faster pace, because of course it has to be commercially viable. And I think Axiom Space has a unique position here, because what we have been doing through these different Axiom missions, as you have seen, by the sheer amount of experiments that we've been doing, is trying to conduct experiments in a repeated fashion to be able to establish baseline proof of concept studies that we can build upon. And actually, Arun's team is one of the prime example here, because during AX2 mission, for instance, already 2 years ago, they had conducted some preliminary experiments that led into continuing their research for their NASA in space production applications grant, which is basically a grant that is geared towards in-space biomanufacturing down the line. So the first phase is showing that microgravity has an impact on the biology that you're doing, a positive impact, whether we are able to make something that we cannot make here on Earth, or it's so much improved that it's now worth doing this in space to make a better product or a perfect product that we cannot make here on the ground. I mean, as you guys know, before even ISS, even during the shuttle programs, there have been so many experiments that have been done. But one issue that we've been having on the science side is, because a lot of people ask me the question, oh, hey, has this experiment ever been done? The answer is probably. But the problem is, because this costs so much, and then you are often doing the experiment only once, it's hard to get them published. Or even, sometimes there is the way that you do the science in space, you might end up needing to use a flight-specific hardware that we don't really have on the ground. So that makes it difficult to compare the results to what has been done previously here on the ground. So what we've been trying to do at Axiom with using our Axiom missions, the private astronaut missions, is to really have a repeated, robust data set that really is publishable, which Arun's team has done. There's several other teams who have accomplished that as well. And then once we have that baseline information, then now, okay, we can scale up or see how we can automate it perhaps, or really dive into it more. Another thing that I really am a big fan of is to try to use commercial off-the-shelf hardware, which is something that, of course, Arun's team has done also during the AX2 mission, because, of course, that makes it not only enables other researchers to do the same as well, but also it helps us compare the results in a much easier way to what we have been doing on the ground, instead of using a flight-specific hardware that only a few people, a handful of people would have access to. So with that in mind, I think in the last couple of years, uh, um, since I started Axiom Space with, for instance, with Arun's team, we've already had done 3 missions to the International Space Station. They have already one paper published, one in review, probably more to come. And this is the story of a lot of our partners. So to your point, Dalen, I think the time is closer than what we think. It's not going to be 10 years from now, but I am hoping that we can come to manufacturing part, at least for some of the projects that we have. Maybe that doesn't require that much of an FDA, like hardcore FDA part, like clinical trials and so on. I think we can come to biomanufacturing on orbit much earlier, maybe in only a couple of years.
Yeah, and thanks for the shout out, Pinar. Appreciate it. But no, this is happening. Like, just to echo what you're saying, Pinar. I mean, part of the impetus here is that the International Space Station is going to be decommissioned by the end of the decade. So we'll need other platforms, other entities to really fill the gap and keep this scientific portfolio going and growing, and without any sort of a gap there. And one thing we talk about a lot here on the Stem Cell podcast is standardization, right? Standardization of stem cell products, standardization of differentiation protocols, because it's just a critically important thing in the stem cell field. And as you're seeing in the space biosciences field, it's also critically important, standardizing the workflows that we're doing for research in space, standardizing the hardware, or in the best case scenario, using terrestrial hardware for in-space applications like what our team, showed a few years ago. So taking I guess a step back, a tangential step, however you want to take this. I mean, you've trained in the lab of a friend of the show, Alison Watry, like you alluded to, and he's been on the show, I guess, a few years ago now. He's pioneered a lot of the brain organoid techniques that are used today, and we've had a lot of brain organoid folks here on the show as well. I mean, how does your training there shape your thinking about, say, like standardization of systems, standardizations of organoid approaches, space biology approaches, and more broadly, how did your training there shape your views on organoids just broadly? So what aspects of that training do you carry forward now, and how did it prepare you for the challenges of biomedical research in non-standard environments like microgravity? And we've talked about this a lot. There's a lot of interesting parallels when it comes to organoid culture in space versus on the ground. Like, for example, a lot of times we're trying to do suspension culture in organoids terrestrially, making them float around, and that's what happens organically in space anyways, right? So talk to us about, your training back then and what aspects of that, have really helped you in your current job.
So basically, what was really useful to me that I learned doing all this organoids or iPSCs in Dr. Alison Watry's lab, helped me in this job is, well, first of all, it helps me working with a lot of different partners. Of course, our own team is one of them, but also we have other teams who are working with iPSCs in other fields as well. We are working with National Stem Cell Foundation, for instance. They're interested in neurodegenerative diseases like multiple sclerosis, Alzheimer's disease, Parkinson's disease. So they're trying to understand how we can make also better disease model and find other or cures, treatments for those patients. So of course, all this training is helping me communicate with other scientists as well. And of course, being a scientist and having done these experiments myself, it also helps me now when we're talking to new partners. These may be people who have never flown before, which is most often the case. It really helps me translate the engineering portion of the microgravity research, which is basically translating your culture on a dish in a hardware, or like putting that in a rocket or doing the experiment on the ISS. It helps me translate the engineering part back to the scientist, and then similarly from scientists going back to the engineering, which is also something that we're doing here at Axiom as well, because of course the one of the main things that Axiom is known for is, of course, making the successor of the International Space Station. Our company is the only company who has the ability to be able to connect directly to the International Space Station. And then eventually we're going to become a free flyer and so on. But that means that we're making our station in a way that we can not only continue the research that NASA and other international partners have been doing on the International Space Station for the past 25 years, continue that research on the Axiom Station, but also we have to be able to do the research in a commercial pace and also have dedicated facilities for research and manufacturing, for instance, because of course ISS has been amazing for doing some of these experiments, but it's not necessarily a very clean environment. It's not a BSL level 2 plus like we have necessarily in the lab, so which is important for organoid research and stem cell biology and things like that. And especially if we're thinking from a manufacturing perspective, we have to have standard protocols, as you were alluding to, Arun, standardization, automation, and all these things. So we are thinking of all these aspects on the Axiom station as well. So having all this knowledge, of course, also helps me get the information back to our engineers as well. So then we have a station that is appropriate to be able to conduct this type of research.
Yeah, I am thrilled by the engineering aspects of this. I mean, again, having my superficial understanding, I'm just kind of gobsmacked by the fact that you're sending stuff up in a rocket. You had me at rocket. So, I mean, the idea of pipetting in space, that's just bonkers. But the engineering and automation, I mean, we talk about that like it's a key part you were mentioning earlier about trying to standardize like same plates, like whatever you can do to enable a comparison to the vast trove of on the ground data, right? Is that's a huge goal, I would say, for standardization purposes. But like the other side of that, which you kind of were just getting at, is all the engineering and tech and automation that you've got to develop, and so that you don't have to rely as much on the human inputs, right? And historically, that's been one of the most convincing rationales for, say, like the space race, right? The moon race was all the tech that spun out of that on Earth, and the knock-on effect, right? So is there a part of that, like, I get it, the focus is how do we do this in this tremendously challenging, harsh environment, but like I would bet a big part of the investment sell, is think about how this is going to change the way we practice science on Earth in our labs, right? Is there any like specific ways that you've thought about that? I mean, you're probably got your eyes like a rune here on getting up there and doing experiments, but I mean, for the rest of us who are stuck on Earth, how is this going to change the way we do business?
So let's say that we're going to biomanufacture stem cells on orbit. Well, we have to think about how we're going to fit that in an environment that, it's going to be much more constrained than what we have on the ground. Because, like, I have been in the center of Cedars Biomanufacturing Center, for instance, where they are doing tons of iPSC production at a time. But of course, in space, even though it's space, but we don't have that much space, or there could be some other constraints that we have to take into account. So one of the ways of thinking about it is, okay, which part of the process during manufacturing or during making a new product in space can really benefit from microgravity, and potentially only making that part in space. So then, we can save some of the space that we have, or like have it in a more smaller environment. Down the line, maybe 20 years from now, I don't know, maybe we'll have huge factories orbiting the Earth. But initially, especially in the next 5 years or so, I think it's going to be more like, okay, what are the parts of the process are really benefiting from microgravity, and just doing that part. And to the earlier point that you were alluding to, like, of course, like the space race has enabled us to have a bunch of products that now we cannot really think our lives without, that were discovered because we had to go to space, like Velcro and things like that, and cordless vacuums. Or I would expect that in a couple of years from now, we're going to have products like that on the biology side, or special medias perhaps, or a way of like doing media handling, or changing I don't know, doing imaging on orbit or things like that. Some kind of technology that will be so much more practical that we have to come up for space, that now everyone else is going to start using it back on Earth. And some of these things are already happening. So I'm very excited about that. So we want to keep as much of the high tech, cutting edge technology that we have here on the ground on orbit as well. But then we might need some tweaking, and then maybe those tweakings might help us get also better products for here on Earth. One of the things that when I was in Allison's lab that we were talking about, and this was, I don't know if he still thinks it this way, but one of the things that he used to always joke about, I mean, half joke, I think, but was because we were using the Cube Labs, these automated systems for brain organoids, and he was thinking of it from a perspective of, okay, so this is something that we're sending on orbit because we don't want to use too much of the crew time, which is a very precious thing. We don't always get a lot of their times, or we don't always get a lot of time on the hood and on the ISS, which is called Life Sciences Glove Box. So we're going to do our experiments in an automated system, which comes with its own constraints. But if we are able to keep the organoids in an automated system, then that could work. One thing that he used to always say is, one day my dream is to have a wall of these Cube Labs and then have organoids in each one of them with different ages, different diseases, and things like that. Almost like a vivarium, you would just go there. Oh, you need this organoid, here it is, go take it. And then, so this was like one of the things that we were trying to optimize for space, but then now it can have an application back on Earth. Like now imagine how much time that would save a postdoc or a PhD student if you were to have like whatever organoid that you wanted at whatever time that you wanted, whatever age. So these are some of the things that we can expect to have in a couple of years, I think.
Music to my ears. And I think music to all of our ears, right? I mean, that's the dream of this field, is to do things in space, not just for the benefit of astronauts or for people going up there, but it's in space for the benefit of all of us here terrestrially. I think that's the dream for NASA, the dream for ISS National Lab, and of course, one of Axiom's visions as well. I mean, we're also very much byproducts of the environments in which we're placed, not just organoids being placed in microgravity, but you being placed at San Diego, right? Of course, you had the ability to train with Allison, and you've been a part of many of the studies coming out of the Sanford Stem Cell Institute at UCSD, led by Kat Jameson, who's actually going to be visiting us, as you know, here at Cedars-Sinai in a couple of weeks. She's actually, a guest of the show as well. We interviewed at her live at ICCR a couple of years ago. I was hoping you could talk a little bit about a study that actually was posted on bioRxiv, collaboration, I believe, with the Mautry Lab and Kat's lab. It was something you were a part of on space-induced neural senescence in brain organoids. I mean, the preprint reports this activation of dormant viral pathways under space conditions. And could you unpack a little bit of those findings for us? What surprised you the most? And what do you think they'll tell us about how stressors like microgravity or maybe even radiation for deep space might affect human neural tissue, whether it's real or, quote, engineered?
Yeah, this was a very interesting study. So it's something that I think so we had sent a couple of brain organoid experiments prior of me leaving the lab, but one of the last missions that I had done in Allison's lab was actually, and this was funny too, because, well, I don't know how much I'm allowed to say about the background of the study, but of course, as you know, Arun, very well, is the space flight is, you have to be very adaptable and very flexible because the flight schedule moves around all the time. So before I left Allison's lab, the last mission that we were doing, that flight kept jumping all around the different timelines. So that was very hard for us in the lab because we kept making organoids at a certain age and then getting ready to load them, but then the flight would get canceled and things like that. So then finally we had a set date, and then we had to prepare organoids, and then we actually had some organoids from this Rett syndrome patient that were in the lab. So of course, Allison's lab is very much focused on the autism spectrum disorders as well. That was kind of one of the big parts, big big disease that we were studying at that time. And Rett syndrome is very interesting because it's an X-linked mutation, MECP2 mutation, of course, and we have shown many different phenotypes of on the brain organoids of coming from these patients, mainly like a disruption of the neuronal networks. I have shown myself on the microglial cell side that there was a defect of these microglial cells in the pruning part of the synapses, for instance, and we've shown several other phenotypes on with other types of cells involved in the brain organoids. But one of the initial pathways that we had seen before even we sent the Rett syndrome brain organoids was that we had a activation of viral pathways, which was interesting because, of course, this is a enclosed system. There's no, there's no virus, there's no nothing. It's in a cube lab. So that was interesting, and we were comparing that data set to what the NASA twin studies had shown at that time back in 2019. Of course, that's the science paper, which was the first, I guess, longitudinal, very detailed study on what was happening in microgravity over a year. So some of the pathways were common in what has been seen in the NASA twin studies with our brain organoids. And then what happened is that once we sent the Rett syndrome brain organoids to space along with the healthy controlled brain organoids, is that we saw that viral activation pathway was even more exacerbated. So, of course, if we go into details of Rett syndrome, there's also a accumulation of single-stranded DNA due to the lack of MECP2 protein, and that has been shown to cause an activation of like interferon pathways, and then kind of like going to the viral pathways. But going to microgravity enabled us to see those phenotypes in a much clearer way. And then what was interested in is that once we had that observation, we started using some antiretroviral drugs to basically inhibit that viral pathway and that single-stranded DNA accumulation. And it's a very cool study because we went all the way to testing those in animal models, and they have worked. And now I think as of in the last couple of weeks, that is now in clinical trials for patients as well. So we're very excited about that because this is a paper that is basically showing that using microgravity, you can see some of these phenotypes in a much easier way, and then now you can translate it all the way to humans, and how you can leverage microgravity essentially to accelerate your research back here on the ground.
Yeah, that I feel like it's not emphasized enough. Maybe just me, but I've been so focused on, because it's like this private commercial idea and we're turning over. It's like, okay, what can we manufacture there? But there is still this huge benefit of knowledge gained and clinical relevance to that knowledge gained, like trying to mitigate the damage that spacefaring does to our bodies. Like, yeah, as you just described there, could amplify or augment our treatment of all these other diseases, right? Because the mechanisms may be so aligned. So yeah, I'm in the space. I love it.
There are also other examples, Dalen. There's one that I would like to highlight because Arun just mentioned Dr. Jameson's work. So they have been flying on all AX missions with us, and what we have been, them flying in all missions not only helped them to repeat their experiments, so it's not one-off, so that now they know that their results are very robust and repeatable, which is something that is somewhat new in the space field as of last couple of years. But what we have seen is that they're interested in how tumor organoids and the protein that is driving their growth, ADAR1, so this is RNA, DNA, and editing enzyme, is affected by microgravity. And basically what we have seen is that the tumor organoids, they grow much faster in space. And also that pathway is very much activated compared to their controls here on the ground. So now in the last couple of missions, not only we've seen that, but we've also tested drugs on those tumor organoids, aiming to really block that ADAR1 pathway. We have used one FDA-approved drug and one that they're actually developing called rebexinib. They are basically, we have seen that even in that accelerated environment of tumor growth, those drugs were able to halt their growth. And actually, the one that they're developing, rebexinib, was working even better compared to the FDA-approved one. So now this is one of the other ways that I'm very excited for us to use microgravity, because not only you can have disease models that develop faster, so in this case, the tumor organoids, but also you are able to do drug screening in a way that even in that accelerated growth, now if that drug is working, maybe you have better chances of that one working in patients as well, maybe with more aggressive types of tumors and like more metastases and things like that.
Yeah, I mean, we talked also while we're, glazing Axiom, this study from Kat Jameson and all as well about like hematopoietic aging, same idea. But yeah, as you just kind of mentioned there, key being that you have these repeat visits, right? You get multiplicity of samples from astronauts, private astronauts in this case, and for just 4 missions to date, the crews of the mission specialists has been very diverse, representing a pretty broad range of stakeholder nations: US, Canada, Spain, Israel, Saudi Arabia, Italy, Turkey, Sweden, India, Poland, and Hungary. That's 11 nations represented amongst 14 individual mission specialists. There were some repeats, right, who went on multiple missions. So the message there is clear, right? The future of humankind in space is not a nationalist thing, right? Is it the same idea among the Axiom crew on the ground? I mean, science is very famously, I know, also famously a bunch of white men in science, but like more and more diverse in terms of gender and people of color. How about at Axiom amongst that ground crew, scientists and non? What's unique about your group? What do you love about working there?
Yeah, I mean, first of all, we have a pretty robust science team at Axiom Space. I mean, science is really at the forefront of our company. It's really the why of everything that we're doing. Our chief scientist, Dr. Lucy Lowe, she is a big pioneer in a lot of the aspects. She's the one that is really driving the message that, science is really need to be, the priority in all of this. And I think this is one of the aspects how Axiom might be a little bit different than some of the other companies. We are very much involved in the science with our partners or with the different countries, helping them from, translating their research or the types of research that they want to do, how we can do that in microgravity, and also not only doing it like once, but what these missions have also helped us, these human space flights, is starting collaborations with different countries, either amongst them, but also with our other partners, like on the in-space biomanufacturing side. All these new nations coming into the space field, so some of them did not even have a space program until recently. They are now really starting this research back in their country as well, and they're very much eager to collaborate. So they were trying to find synergies between these different countries, or even with our partners here in the US. And this is how we're going to advance science much faster, I think, and using different partners' resources and then strengths and so on. And it's really a whole new level of collaboration, I think. And that's something that I'm really excited about. And that's, I think, puts us in a very unique position. And we're very excited about that. One other thing that I wanted to mention is kind of a side note, but because you said that we was a very diverse crew from all the Axiom missions, and of course these are like from different nations. Not only that, but we're also very much focused on trying to enable access to microgravity to people who may even have medical conditions that were initially right off the bat kind of removed from the cohort. Like, for instance, people with diabetes. One of the things that we have done on AX4 is actually send a glucose monitor to be able to see how, and also like insulin and so on, to see how that can be stable, and how we can monitor the glucose levels and things like that. All of it to as a preparation to one day hopefully send someone who has diabetes to an International Space Station without risking their lives or anything like that. So it's really, we're trying to open the doors to anyone in microgravity.
Space for everybody. That's the vision, right? And space is for everybody, not just for the folks who can afford it or for the folks who have been selected as the best of us traditionally, as NASA astronauts have been, right? But that's the dream for the future of of space flight. Is we want everybody to be able to access it, not just the elite when it comes to human fitness, but folks who have disabilities, folks who have underlying medical conditions. And what you guys are doing at Axiom is opening up space to all of us, not just folks from here in the United States, but like we were talking about, astronauts from all over the world. And we've been really fortunate to work with a number of these folks. So thank you so much, Pinar, for sharing your vision, sharing Axiom's vision for the future of space. And as you alluded to, space is really the most collaborative endeavor out there, at least especially when it comes to doing science in space. You need, of course, incredible cell biologists, biomedical scientists, but also engineers, aerospace engineers, mechanical engineers to make these projects happen. And I think there's a lot we can learn in the stem cell field for in terms of doing science in space and translating some of those findings and some of those workflows to terrestrial applications, right? So there's a lot we can learn from what you guys are doing. And just on a personal level, thank you, of course, for being such a great friend and collaborator over the years. We honestly couldn't have done this without you. So before we let you go, we're going to ask you a couple of peripheral questions just to help our audience get to know you a little bit better, like we always do. So first up, if you were not at Axiom, and broadly, if you were not a stem cell biologist, which you still are, what would you be doing with your life?
Well, this is, I think, something that I started really thinking hard when during COVID, I was writing a lot of papers, and we were sending a lot of papers for submission. And how you guys, obviously everyone who also listens to this knows how this goes. You always have to find like the right way of like responding to reviewers, and you kind of make your case. So I think one of the things that I would have liked to be instead of like if I wasn't a stem cell biologist, is a lawyer, either a lawyer or a judge, because sometimes I did get frustrated with the editors, decisions or things like that. And I was like, oh, but we have all the evidence here. Like, what is happening? Like, how can you not be able to make the right judgment call here? So that's probably one of the things that I would have gone to. And also, my dad used to be a criminal lawyer, so I was like, maybe I have part of his like genes in me about that. But yeah.
I like that. I don't think we've ever gotten that answer before, lawyer, but that makes a lot of sense. I personally thought you were going to say sailor because I know you like to sail down there in San Diego, but you can do both, I suppose. Last question, and you've already given some advice already, a lot of advice already on this show, but what's the best piece of advice that you've ever been given, professional or not, especially geared towards the trainees listening here?
Yeah, I mean, I think really networking is like sometimes even like it can be even more important than your skills as a like scientist. It's really, I think, important to nurture those relationships and things like that. Like just overall, like be nice to everyone, and kind of like these like life things that are transferable to anything, but also in this field. I would say, of course, this field is very fast paced and it's ever changing. Being adaptable is going to be really important. Things get canceled or, move around a lot. Of course, it doesn't mean that no one really gets frustrated or anything, but being resilient, I think, is really, really important. Being brave and, adaptable. It's going to have a lot of challenges, but, don't be afraid. I think that's part of the fun too. Or even if you're afraid, just, continue persevering. And yeah, perseverance, resilience, all Mars rover names that we can think of. I think they're also important here. And yeah, just overall, I think just as long as you're consistent and then you're perseverant and hardworking, I think things will work out. And if you're interested in the space field, I think it is now time for more and more people like me to start transitioning to this field. And we will need more and more, especially with all the commercial companies coming into this field, all the other pharma industries and other industries coming in. We're really going to start needing more and more scientists, because once we're going to have the stations, well, we're going to start doing all the science. So this is where we're going to need all the scientists and also scientist astronauts. So then they can do all these specialized, very high-tech experiments on orbit as well.
Wow. I mean, it's funny because you're listening to all the attributes, what you need. And I got kind of confused there for a minute. I was like, wait, is she giving advice on how to become an astronaut or how to become a scientist? And I realized by the end that it's both. You're usually scientists, astronauts. And in fact, that line is blurring, I think, in large part, thanks to the initiatives and the passion of people like yourselves and Arun. I mean, you say, Arun, science is for everyone. Science is not, space is for everyone. Space, it's not for me, you guys. I grew up in New York. I've learned to live without space so you guys can have it. But I'm sure there's plenty of our listeners that are into space. So get after it, y'all. Like Pinar said, it's coming. Thank you so much for this conversation. Really eye-opening and a lot of fun. We'll have you back on again.
Thank you both so much for having me.
All right, you guys, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. They were all in Cell Stem Cell, so you don't even need a link. Just go to Cell Stem Cell and look down the page. If you enjoyed this episode, please feel free to share with a friend, colleague, or on social media. You can also reach out to us on X at Stem Cell Podcast or Blue Sky at stemcellpodcast.com or via traditional email at info@stemcellpodcast.com with feedback or to suggest guests.